defined as a chemical messenger. It's made at one location, exerts its effect at a distant location within the body. Now, when we talk about the endocrine system, there are many organs in the body that have endocrine function. Some of them just, that has a major, maybe an exocrine function or some other functions, just a few cells in it that will have endocrine function. And so when they talk about the endocrine system, they'll put up a bunch of different glands. We could have put up more than this, actually. Heart actually has some endocrine function to it as well, believe it or not. And so, you'll see in terms of when they put up a figure like this endocrine system, sometimes they'll try to include everything, sometimes they'll only try to include small things. For instance, we had neuroendocrine cells in the respiratory system yesterday. We're going to have the same type of cells, enteroendocrine cells, which are a type of neuroendocrine cells in the GI system. You don't see that up here, do you? You will see that on other images. Now, when we are going to cover the endocrine system, the ones that we are going to cover, the ones that I've got in red. We're really not going to cover the hypothalamus, although you talked a good bit about the hypothalamus with Dr. Olgan, didn't you, when you talked about the function of the endocrine system. We will spend a good bit of time on pituitary. We'll move on to the thyroid, the parathyroid, and the adrenal gland. Thymus we've already covered. We did the lymphoid organs. Pancreas will wait until we get to the GI, since it has major exocrine function with the GI. And then ovary and testes, great deal of endocrine function there. We'll wait till we get to the reproductive systems for those. And kidney has some cells in it that have endocrine function, but it's primarily involved in excretion of nitrogenous waste, correct? And we're talking about sodium, potassium balance, and so on, but that's not the main function of the kidney, so we will cover that with the urinary system. And so we're not going to cover all the components of the endocrine system in detail, just the major organs that are primarily endocrine in terms of their function. We'll pick up most of the others. Either we already have, like the thymus gland, we'll pick up most of these other ones in other systems of the body here. Now, you know that endocrine function is related to cells secreting into a capillary bed. Okay, now technically they don't have a mechanism where they put things from inside the cell directly in the capillary. They secrete their hormone into the connective tissue surrounding that capillary, and it then has to get in the capillary. And that is why you will often find fenestrated capillaries within the endocrine organs. Not always, but many of them will. And there's a few that even have sinusoidal capillaries in there, just making it easy for large, maybe protein molecules. If that hormone is a protein, make it possible for it to move in and out of capillaries a lot more easily, in this case into capillaries a lot more easily, by making those capillaries extra leaky. Now, when we look at it here, terminology that you're always going to see when they first start talking about how endocrine organs are arranged histologically, they talk about clumps and cords. Okay? I'm not going to point on an exam and say, what is this? Is this a clump or a cord? It doesn't jump out at you as clumps or cords. It really has to do with how the blood vessels are running through it. For instance, if we look here, here's a capillary running. Here's another capillary, so you've got two almost parallel. Can you see a cord of cells right here? Okay? So in other words, you can make that out. On the other hand, this down here really doesn't look like a cord. We'll call that a clump. Okay? Cords and clumps don't jump out at you, and you're going to see some, you read like parathyroid gland, and we'll talk about how this is typical cords and clumps, and you may look at the image you're looking at and say, I don't see it. Okay? That's about as good as you ever see a cord and a clump. Don't worry about it. Okay? I only put it in there because it's, like I say, every website, every textbook talks about cords and clumps being your typical endocrine histology. Okay? Some places it will stand out, particularly cords when we get the part of the adrenal gland. Other places it really doesn't. And the clump is, you can't even really define what a clump is. So, but you won't necessarily see that. It doesn't jump out at you when you take a look at that. What we are going to spend a little bit more time on, because it's important, is talk about staining properties. And we did this with epithelial tissues, and most of these endocrine glands are epithelial derived. But we need to talk about staining properties. You know that you've got different types of hormones. There are protein And then if it's only a few amino acids in length, it's a peptide, but they're basically going to be made the same way, correct? Amino acids linked together on rough endoplasmic reticulum. Okay? You also have steroid-based hormones, and you've got catecholamines. Well, let's talk first here about the protein, and I'll include with that peptide stage hormones. If you have a lot of rough ER in your cytoplasm, we know you're going to pick up the hematoxylin, correct? The ribosomes have RNA like DNA, where the nucleus picks up the hematoxylin, so it's going to make a purple staining cytoplasm. And we look in here. Here's a series of cells right here that are protein secreting. Okay? They've got darker purple staining cytoplasm, which is due to the rough ER that's in there. Okay, now you've also got a potential paradox coming in, because they're also going to have a lot of secretory vesicles containing a protein hormone, correct? And proteins, depending on their amino acid makeup, may not pick up much stain to speak of, or they may pick up a lot of hematoxylin, or they may pick up a lot of eosin. Okay, so if they pick up a lot of eosin, what's going to happen here is once you've got a lot of protein built up in those secretory vesicles, rather than a protein secreting cell staining dark with the hematoxylin, it's going to stain pink with the eosin. And that's what you've got right here. We're going to see this in the adrenal gland, in the pituitary gland here in just a little bit. Both of these are protein secreting cells. Okay? The basic foundation of the protein secreting mechanism is going to stain purple, okay? Whereas if it's happened to making a lot of a protein that picks up the eosin, it will stain pink, okay? And so that's what we can see both of those types of cells here within the pituitary gland, the adeno hypothesis. Okay, steroid secreting hormones, those cells are going to stain a little bit different. If you're a steroid secreting cell, you don't have a lot of rough ER, that's for making protein. Instead, what you got in a steroid secreting cell is a lot of smooth ER. The other thing you're going to have, because you're making a lipid, a cholesterol, which would be a lipid, you know that lipids are very high in energy, correct, in terms of energy storage. Well, if you're making it, that means you've got to have a lot of ATP to make it, correct? If you have to have a lot of ATP, you've got tons of mitochondria. Both mitochondria and smooth ER are internal membranes in the cell, and those pick up eosin, which means it's going to stain pink. Okay? Now, the other thing, once you've got all these little vesicles containing the steroid hormone, what happens to lipids like steroids during fixation? You remove them, correct? And so what you see at the foundational level here, you see pink cytoplasm, and if you look carefully, you can see little tiny empty secretory vesicles, can't you? Okay, this is probably a little bit overstained here to get some of this pink color, but you can look in here and you can kind of get the idea. Yeah, I've got just barely making out, if they show up really well down here, if I look other places, I can get the idea. I've got these little tiny, what look like pink, like clear air bubbles in there that are the secretory vesicles from those steroid secreting cells. Now this has come from the adrenal gland, and the cytoplasm here doesn't stain so pink in terms of a little bit different. It picks up a little bit of the hematoxylin as well, but look at all those secretory vesicles in there. Okay, when you see that many air bubbles, so to speak, in an endocrine organ, you know that you're making a steroid-based hormone. Okay. All right, let's move on and talk about the pituitary gland, or the hypophysis is how it is sometimes referred to in older textbooks. It's located on the floor of the brain and hangs down, sits within the cella turcica that we've looked at previously. You learned with Dr. Olga, it's sometimes referred to as a master gland because its secretions control other endocrine glands as well. It's part of that feedback loop that allows you to control endocrine function of various endocrine organs. In order to understand its histology, it's important to understand its development. That's one thing I like about your textbook. It actually has some developmental images that are better than most embryology textbooks. And this is one that is true here as well. We get to, later on in the semester, we get to the formation of the brain. This is a stage where the brain is a neurotube. It's a hollow tube at this point, and the nervous tissue is, on the outside of it here. You also have the folding of the embryo where you form the primitive gut, okay, which will literally go from the mouth to the anus, or what will be the mouth and what will be the anus, okay? And within what will be the roof of the mouth and the base of the brain, you've got tissue. This is the blue is the nervous tissue. This is the lining of the mouth. It's going to pouch out from the brain, pouch down, from the roof of the mouth, pouch up, and they're going to come in contact with each other. And that's just as you're starting to form the bone of the hard palate, which will then separate that lining of the mouth from the lining of the mouth and put it up within the brain. And so that what you end up having here is a pituitary gland that is part nervous tissue and part epithelial tissue. Okay? And I'll point out one thing we'll come back to a little bit later. This part right here is Rathke's pouch in terms of as that epithelial lining of the mouth pouches upward. It's got the Rathke's pouch. This is referred to as a remnant of Rathke's pouch or vestigial of Rathke's pouch. Some books will just still call it Rathke's pouch, but you can see Rathke's pouch right here still, or at least the remnant of it, can't you? Okay, we'll often see that. We'll look at it underneath the microscope as well and realize at this point, your oral cavity is down here, and so you've got bone separating, bone being the sella turcica now, separating the epithelial portion from where it was derived here from the oral cavity. All right, now let's look at it on the slide here. We said it's got a nervous system component and a epithelial tissue component. Here it is in section. If we take a look, here is the bone of the skull. This is the sella turcica right here that it's sitting in. Here is the pituitary gland. If we take a look, which is the nervous tissue and which is the epithelial tissue, well, if you look up here, here is the infundibulum that suspends it from the base of the brain. Here's the optic chiasm. Here's the hypothalamus. Here's the ventricle. I'm going to say third ventricle if I remember my brain anatomy correctly. And so all of this, kind of the pinkish color, is nervous tissue, which says this must be my epithelial tissue right here. Okay? And so you can get the idea that, yeah, part of it is nervous tissue, part of it is epithelial tissue. And of course, the hypothalamus controls a good bit in terms of the function here of the pituitary gland. We'll look at that here in the next slide. All right, we look at the relationship between the hypothalamus and the pituitary gland. It differs in terms of, is it the nervous system component or is it the epithelial tissue component? And I've divided it here into anterior and posterior pituitary. That's what you're used to dividing it into. We're going to divide it up a little bit different histologically, and we'll see why we get here. But in terms of, let's take first the anterior pituitary. You described this. You probably had a very similar image to this with Dr. Olgan. In the anterior pituitary, what you've got is a portal system. Portal systems are when you have two capillary beds in series. Normally you go artery, arterial, capillary, venule, vein, and back to the heart, correct? In a loop from the heart back to the heart, you only go through one capillary bed. A portal system is when you have something that goes like artery, arterial, capillary, either venule or arterial, capillary bed again, and then venule vein back to the heart. You've got two capillary beds in a loop from heart back to heart. And whenever you have a portal system like that, it's a specialized transport system. For instance, you guys you may have talked about this one. In fact, you should have. You talked about one in the circulatory system We had capillary beds in the intestines that fed through a vein that came to other capillary beds in the liver before going back to the heart. Okay, we'll look at that one again as well. But basically what you've got is you've got arterial blood coming into the hypothalamus. Hypothalamus can then put releasing hormones or inhibitory hormones that it will put into this capillary bed. It will then come down in from this capillary bed here in the anterior pituitary, either stimulate or inhibit cells here of the anterior pituitary from secreting their hormone. Okay, you talked about that, but that's what we're talking about here. It's a specialized transport system, so that rather than these hormones here that we're producing up in the hypothalamus, rather than just putting them in circulation where they are diluted down through all the blood of the body, instead what we're going to do is have a specialized transport system, so they are very concentrated here where they're just put in, and they're very constant, they're in the same concentration down here, because we haven't diluted it down with all the other blood of the body. Okay, makes sense? Okay, nice little design feature we have here to allow very intimate communication, very fine controlled communication, chemical communication, I might add, endocrine communication between the hypothalamus and the anterior pituitary. Posterior pituitary is a little bit different. It's nervous system in terms of its component, and there are two nuclei up here within the hypothalamus. And I would make this drawing a little bit different. I'd put cell bodies up here. I mean, I don't like the fact they've got it branching and coming in. I'd put cell bodies up here. Here's cell bodies of neurons, and their axons reach down through the infundibulum into the posterior pituitary, and I put the little synaptic bulbs down here. Okay? These are functionally, in terms of how they work, neurons. Okay? They make a neurotransmitter up here within these cell bodies, within these two nuclei up here in the hypothalamus. They transport it down their axons, just like you talked with Dr. Olgan about how neurons transport down secretory vesicles of neurotransmitter down the axon. It collects the little secretory vesicles now in the synaptic bulbs down here. The difference is that when there's an action potential causing the release of that neurotransmitter, it goes into capillaries. And so because it goes into capillaries, it then acts as a hormone. Okay? And so it's kind of a nice little hybrid, isn't it? It's made and transported and released just like a neurotransmitter, but because it's not going on to another effector, a secondary neuron or a muscle cell or something like that, it's being released into the bloodstream. It's now going to act as a hormone. Okay, so very different here. Here you're going to have a lot of cells. You're going to have a lot of capillaries. I don't show the capillaries here in the posterior pituitary. A lot of capillaries in both because you're going to be secreting hormones in those capillaries, correct? The difference here is that we're going to see over on this side, in the anterior pituitary, we've got a lot of cells there because we're going to stimulate cells to release hormones. On the other hand, over here, we have cells making the hormones. Where are the cell bodies that made those hormones? They're up here, aren't they? And so we're going to see a lot of axons passing through here. There'll be some supporting cells, but it's not going to look even close to the same. One's nervous tissue with a lot of axons and supporting cells. The other's epithelial tissue, where it's going to be highly cellular. Okay, any questions there? Yeah, no, it's not important, and I know what the DO note, those have the DO program, I've taken that out because they're named different things in different sources. Okay, and so yeah, the nuclei that are named up here and what I've got in the notes are different, and I didn't realize I haven't taken them out here like I have in the DO course. Just look, different sources have different names, and particularly when I am doing something that's not my area, naming these nuclei, I tend to use the old names that aren't used anymore because I'm not up to date, but they've changed. All right, now the epithelial portion of the pituitary gland we're going to see is not equivalent to the anterior pituitary. Okay, some histology textbooks, newer ones, will talk about it in terms of anterior and posterior pituitary, but that's not quite accurate. We're going to see, and I'll get to that at the very end here when we talk about the pituitary gland. The technical name for the epithelial portion of the pituitary gland is the adenohypophysis. Okay, I used that term earlier. I'm now defining it. Adenohypophysis is the epithelial portion. Neurohypophysis will be the neural portion of the pituitary gland. And I'll come back and make correlation once we talk about subparts of each of these. I'll come back and make the correlation between anterior and posterior pituitary. All right, the largest component of the adenohypophysis is what we call pars distalis. Okay, it's the anterior portion here of the pituitary gland. It's really what we normally would refer to as the anterior pituitary as well. We'll see that in just a little bit here, though. Histologically, it's what we looked at earlier. The example I used, the cords and clumps, was from the pars distalis here. It turns out when we look at it, there are three visible types of cells that stain. If we look over here with H&E, you've really got to, get it just right. Stain, destain, stain, destain to get it just right to see these cells really well like you have in this image. You've got stains which have dark staining of cells that have dark staining cytoplasm. You can see little granules in there. We're going to refer to those, they have basophilic cytoplasm, and so we're going to refer to those as basophils. No relationship to the basophils, the white blood cells named basophils. Okay? Only relationship is they both have basophilic cytoplasm. Now the basophils is basophilic granules. Okay, but no relationship in terms of their origin, function, anything like that. They get the same names based on their staining properties. Okay, and then we have some that stain a little bit more pink in here. They have more acidophilic cytoplasm, so they're referred to as acidophils. And then we've got some that really don't pick up much stain. We call those chromophobes. Now, like I said, with H&E, you've really got to work to get that so it shows up this perfectly, okay? And even then, it can be subtle. I guarantee if I put this up the first day, and I told you there's 3 cell types in there, you'd have trouble finding the 3 cell types, wouldn't you? Okay? Now your eyes, I think, are a little bit more discriminating you can. What's often used is an azan stain. Because an azan stain with that, we get these basophils, they still have that darker purple cytoplasm to them. The acidophils stain more of a yellow rusty color, okay? And then the chromophobes, they're all down in here, they aren't picking up any stain, okay? So the azan, with the H&E, our standard stain over here on the left, it's very subtle. You gotta look carefully to see the differences, and you've gotta have it stained just right. Azan, you can be a lot sloppier in terms of not needing to stain, destain, stain, destain to get it right, and you can still distinguish the 3 cell types in here. All right, let's talk about these 3 cell types here a little bit. Chromophobes tend to be small, but not always, but they do tend to be smaller than the others. Cytoplasm doesn't pick up any stain, and because of that, you don't get, it's hard to sometimes see where it stops and ends. As a result of that. We don't know what chromophobes do, okay? We know that they're currently not doing anything, and depending on what source you look at, they'll hypothesize that these are old, worn-out cells, or it might be that these are acidophils or basophils that are just taking a break. It might be these are reserve acidophils, basophils. I haven't looked in the last couple of years to see what the latest theory, or if any one of those theories has gained more traction than the other. Not that important, just identify them as chromophobes and don't have any known function for them per se. All right, the basophils, oops, let me take the order I've got them. The acidophils, they're going to be picking up the pink eosin there in H&E. There are 2 subtypes. We can't tell them apart unless we have an antibody staining for the product. There are somatotrophs, which produce somatotropin. You probably learned it as growth hormone. There is another population that are mammatrophs that produce prolactin, and prolactin is involved in milk production. There's probably other behavioral patterns associated with its secretion as well, but I won't go into that per se. The basophils, the ones that pick up the hemotoxin in their cytoplasm, three subtypes here. There are thyrotrophs that produce thyroid-stimulating hormone. There are gonadotrophs that produce LH and FSH. And there are corticotrophs that produce adrenocorticotropic hormone, or ACTH. Okay, now the other part of the adenohypophysis is what we call pars intermedia. It's kind of a wedge shape of tissue in between the adenohypophysis over here and the neurohypophysis here. And in between the adenohypophysis, or in between the pars distalis and the pars intermedia right here, there will be little remnants of Rathke's pouch. And sometimes those remnants will contain little material in there. When you contain material inside an opening like that, a follicle like that, we refer to it as colloid. That's just a general term, okay? Material in that little opening. But these basophils right here, I say basophils because they're really picking up the hemotoxin, that makes up our pars intermedia. And I'll show you some in lab where you get a little bit more of the view of the pars distalis and pars nervosa on each side. And it kind of does look like a little wedge here of basophils in here. Now that pars intermedia, what do those basophils do? They synthesize melanocyte-stimulating hormone, MSH. I'm not certain if you talked about that very much with Dr. Olgan, but it stimulates cells of the body to make melanin, which is our major pigment of the body. And that determines your natural pigmentation of the skin, your ability to get a suntan versus a sunburn, okay, and so on. There's probably also behavioral aspects of this that are often, at least mentioned. Why? Because it is very prominent in animals. You think of that bird around here. Dr. Mahalik is a birder. Now, he'll tell you how many thousands of different birds he's seen, species he's seen. There's one that's way up high in trees here called the scarlet tanager. When you see it in mating season, the male is bright red with black wings. You see it right now, he's kind of this rusty yellow brown color. Okay? What happens is during mating season, his melanocytes go into full bore to make him that bright color, and he's a lot more territorial then. You can play his call on your cell phone, and he'll come find you in the woods. Okay? Thinking there's another male, and I'm the male in this area. Okay? Basically, it's associated with mating behavior and behavior within animals, and so they often want to say there may be behavioral aspects of MSH as well in humans. Okay? And so when they say that, they're extrapolating from animals. I don't know they've ever shown any true behavior, but you'll often see that, as a possibility at least in man. Okay? The other part that we need to at least mention here, because it's epithelial origin, is called pars tuberalis. We look here at the infundibulum, the stalk that suspends the pituitary gland from the hypothalamus above. It's got a core of those axons that are coming down from the hypothalamus, but it's got a wrapping on the outside of epithelial tissue, and that wrapping is referred to as pars tuberalis. No secretory, no endocrine function associated with it, though. Okay? Now, the neurohypophysis is composed of the core of the infundibulum right here. A lot of times they just refer to it as the infundibulum and then a covering of the infundibulum of pars tuberalis. So whether you consider the whole thing the infundibulum or just the core, it doesn't really matter. But what we're going to look at and spend a little bit more time on here is our pars nervosa down here. That pars nervosa, you've got in the order of 100,000 or so axons coming down. This is what pars nervosa looks like. You look at that, doesn't jump out at you and say, hey, I'm pars nervosa, does it? Not real distinctive. You don't see a whole lot of cells. You've got cells in here that are accessory cells, supportive type cells in here. We call pituocytes. Cells of the pituitary is what it describes. But what you primarily have at axons coming down, and you kind of get the idea, like right through here, there's something coming down through here. That would be a bundle of axons coming down. Kind of get the impression right through here. I've got something passing up and down through the screen here. That would be a bundle of axons as well. You can see capillaries in here. Right here, I'm in the middle of a capillary. What you can also see in here, and I'm not going to make you identify it, but you're going to see it. It's labeled here. You're going to see it labeled other places, are what they call herring bodies. Herring bodies are simply the swollen synaptic buttons at the end of axons. And sometimes they can be so full of that neurotransmitter slash hormone that they show up as something like this. Okay? The issue, one reason I'm not going to make you identify them is a lot of times that could be a capillary, and if you had a microscope, you can find focus up and down to identify, distinguish between the two. With the static image, sometimes it's very difficult to say, you know, they'll label it a capillary, and you say it looks like a herring body to me. That's because they could find focus up down and say, yep, that's definitely a herring body, not a capillary, or vice versa. Okay? And so that will look a lot alike. Don't get caught up in terms of, I thought that was a capillary, but they're calling it a herring body. They look very similar. They can look very similar. Like I say, with fine focusing, you'd be able to distinguish between the two, though. Okay, what are the hormones released? Two hormones, antidiuretic hormone and oxytocin. Okay, there's two hormones made up within the hypothalamus, released through pars nervosa, though. All right, let's talk about the correlation here between anterior and posterior pituitary. Looking at how it's formed, this remnant of Rathke's pouch, it can be seen, in fact, you can see it in this drawing right here. Okay? And when they were first, with the gross anatomists looking and saying, we've got anterior and posterior pituitary, what they did was slice through it with the naked eye or a microscope, could look at it and say, there's slightly different colors to it, and they could see the remnants of Rathke's pouch. And so everything behind Rathke's pouch they called the posterior pituitary. Everything in front of Rathke's pouch, or the remnants of Rathke's pouch, they called the anterior pituitary. Where is pars intermedia, though? Red is epithelial tissue. Pars intermedia is posterior to Rathke's pouch, correct? And so pars intermedia, even though it's epithelial in nature, is part of the posterior pituitary, correct? All right. And then you've also got the infundibulum, pars tuberalis, that really aren't part of anterior or posterior, that are nervous tissue and epithelial tissue. Okay? And so that's the distinction here in terms of why histologists talk about an adeno and a neuro hypothesis compared to an anterior and posterior pituitary. It's mainly the pars nervosa, the pars intermedia, that gives us the issue. It's epithelial in nature, but it's part of the posterior pituitary. Okay, does that make sense to everybody? Rathke's pouch, or the remnants of Rathke's pouch, is the dividing line between anterior and posterior pituitary. Pars intermedia is on the wrong side. Okay? Once again, some websites, some textbooks, newer sources typically, they refer to as anterior and posterior pituitary, and that's not 100% correct because, we're talking about anterior pituitary as epithelial tissue, and they may hedge. Posterior pituitary is primarily nervous tissue. You got to hedge a little bit on that because it's not the clear distinction between anterior and posterior, between nervous tissue and epithelial tissue. All right, any questions there? All right, let's move on then and talk about the thyroid gland. Gross anatomy, I'm thinking that was Dr. Engstrom. Thyroid gland, kind of a butterfly-shaped gland wrapped around your trachea, the anterior cervical region. If you were to take that butterfly off the trachea and look at the back side of it that's against the trachea, what you're going to see is four little glands. I'm sorry, I'm getting to the parathyroid gland. Here's the thyroid gland right here. That thyroid gland, if we look at it, low power histology, low power magnification, about magnified 12 times, what you see looks like little pink islands with cells around them. I grew up in a little town in Central Florida about the size of Blacksburg. It was known as the city of 100 lakes. And you fly over, you see all these lakes, and you see houses around the lakes. That was preferred real estate to be on the waterfront there. This reminds me, except the lakes weren't pink, they were blue. Okay? Bunch of little lakes with the nuclei being the houses around the lakes. Okay? That's the histology of the thyroid gland at a very low power magnification. If we go to higher power magnification and look at it, here is one of those lakes, and this is the functional unit here that we refer to as a follicle. A follicle consists of follicular cells, okay, the nuclei around the edge of the follicular cells, and then this region in the middle here, we said when we talked about it with Rathke's pouch, we said we call it colloid. That's just the name of the material in the follicle. If we want to name it specifically, its chemical makeup, it's thyroglobulin. Okay, and we'll talk about thyroglobulin here in just a little bit. Now, these follicular cells are going to be a simple cuboidal. And the terms they often use here is it a high cuboidal or a low cuboidal. These right here would be a low cuboidal. They're slightly shorter than square if you draw lines in between them here. And what that says is these cells aren't very active. They aren't very active because they filled up their follicle already, and they're not taking out of that follicle or putting into that follicle at this time. If they are more active, that they're adding to the follicle or taking out what's in the follicle, or taking out of the colloid or putting into the colloid, they're going to be a tall cuboidal, a little bit slightly more than square, slightly taller than square in terms of the shape. And so you'll often see that the textbooks and websites talk about low cuboidal, high cuboidal when we talk about a thyroid follicle here. The colloid is made up of a molecule we call thyroglobulin. This is more detailed than you have to know. This is more like something Dr. Olgan might have shown you. But the take-home lesson from this is inside these follicular cells, you start to make thyroglobulin like A, convert that to B, convert that to C, and then you put it out here in the colloid. And from there, if that was C you put out there, you convert it to D and E, maybe F. And then when you need thyroid hormone, you take that finished thyroglobulin back out of the colloid and put it inside the cell, and you convert it to G and H would be T4 and T3 you learned about that will be secreted into the bloodstream. What is thyroglobulin? It's an intermediate form of thyroid hormone. Why are we doing it this way? Well, it makes it so that rather than having to go, okay, interpituitary says we need more thyroid hormone produced, rather than going A, B, C, D, E, F, G, H to produce it, we've already got it all the way at F, don't we? So all we gotta do is take it up and just go GH, and we can make it more quickly. Okay, does that make sense? Okay. Don't necessarily do that with other hormones of the body, but we do it here. All right, now if we talk about the follicle as being the functional unit of the thyroid gland, it's where you make thyroid hormone that controls your basal metabolic rate under the control of TSH from the anterior pituitary. You know all that from last semester. Turns out there is another endocrine function here, and that is calcitonin that controls blood calcium levels. Here are the follicles, okay, in terms of here is the colloid. It's sliced thin, so that maybe this one looks pink, this one does not. Sometimes the colloid can completely fall out of the follicle, so it looks white. But basically, if these are the lakefront homes, so to speak, in between, little tiny branches of tissues in between, little wedges in between those follicles, sometimes there are secretory cells in there that we call C cells or parafollicular cells. I've only got them listed as parafollicular cells, but they're often called C cells as well here. And those are the cells that will secrete calcitonin. Calcitonin is going to be released in response to high blood calcium levels. It will increase osteoblast activity, so that you're getting calcium out of the bloodstream, putting it into bone. All right. Now, the next gland to talk about is the parathyroid gland. We said that the thyroid gland is butterfly-shaped, wrapped around your anterior trachea. If we take that butterfly off and look at the wings, the part that were up against the trachea, we're going to see four little pieces of tissue that we'll normally see when we talk about embryonic development, if things have migrated properly. Four little pieces of tissue there that are the parathyroid glands. Okay? Right and left, superior, inferior, the four different parathyroid glands there. If we look at this low power magnification, magnified 40 times, hopefully you look down here and you recognize that now as thyroid gland. Nothing else really looks like that, so even low power you can distinguish it. Wish they didn't have an L in the middle of this. A lot of dark staining nuclei. What do we have? Lymphatic nodule, don't we? Can show up anywhere. Just infection of the thyroid gland. There's one there, okay? Guaranteed, because we've got a lymphatic nodule. You don't have to know per se, but that just brings in the other lecture. And then we've got a little bit of tissue separating thyroid gland from this over here, which will be that parathyroid gland. Close association, so we're always going to see a little bit of separation from it. And if we look at it, that is going to be a capsule separating thyroid and parathyroid. And it does form trabeculae that come up and partially divide into lobes, not real well-defined lobes necessarily, lobes here of the parathyroid gland. If we take a look at the parenchyma, the middle portion, the functional portion of it here, we've got two different cell types in here. Low power magnification, you make out there are cells down here that are pink and they're large, fairly large cells, or if you look up here, you've got cells that don't pick up much stain, but they're small because the nuclei are so close together, correct? So that tells me that the cells are very small. Let's go to the higher power magnification. We can see capillaries in here, as well as down here. That's what the bright red is the blood in the capillaries. You're going to have plenty of fenestrated capillaries in there. Got to be adipose. We'll come back to that in just a little bit. But these cells up here, these tiny cells that don't pick up much stain, those are our chief or our principal cells. Once again, very small. That's why nuclei look, they're so close together. Very little staining to the cytoplasm, but it turns out these are the functional cells here. These are the ones that make parathyroid hormone, which is the antagonist to calcitonin that we just talked about. Okay, we'll stimulate osteoclast, which then will raise blood calcium levels. All right, what about these pink cells? These pink cells, it turns out, aren't there at birth. Begin to show up, say, puberty or maybe a little bit before puberty there. And when we look at them in terms of why are they pink, they are chock full of mitochondria, which we can barely see if we have a good light microscope and tune it well. Use an electron microscope, we can see them very well, chock full of mitochondria. We've got these cells that appear later on, say just prior to puberty. Chock full of mitochondria says they've got to be doing something, but we have no clue what they're doing. Okay? Don't appear to be secretory, but really don't have any function ascribed to them. I haven't looked in the literature in the last couple of years, but probably 2-3 years ago I've looked at and can't find anybody really giving a good, solid, evidence-based, here's what they do. Okay? And so, got to be doing something. You don't have cells with all these mitochondria just doing nothing, but we don't know what they're doing. All right, and those cells, I didn't give a name to them. They're oxytros. Some books will call them, some sources will call them oxyphils. Okay, oxycells, if you want to think about that. Oxytros or oxyphils will be the name. They typically will occur in clumps. Okay, they're all clumped together here. Sometimes you may spot an individual one. I don't here. We'll see it in lab. Well, here's a newer clump of them right here. Okay, and that clump will grow over time as those cells will continue to divide and develop. The other thing to say about this is we'll accumulate adipose tissue in here with age. It's labeled as A, all of this is adipocytes. Here you can see higher magnification of those adipocytes. It could be up to about half of that gland in grandpa. Okay, and no known function in terms of more adipocytes in there, no known function, any reason particular why. All right, the last gland to talk about here will be the adrenal gland. And the adrenal gland, or the suprarenal, superior to the kidney is what suprarenal means. Flagged organs, and man, they rest right on top of each kidney. If you dissected like a cat, they're in close proximity, but they aren't touching each other. And so if you read, for instance, one of the websites I've got for you is from VetMed. There they may not talk about it sitting on top of the kidney, because I don't know where it is in a dog. I know where it is in a cat, because I've dissected enough cats. That it's not necessarily in contact with the kidney like it is in man. Functionally, as well as histologically, it's often referred to as two glands inside a single capsule. Okay, why do we talk about it like that? Because you can see, whoops, already done that. You can make out there's different portions to it here. Turns out we've got an epithelial portion here wrapping around a nervous portion in here. Okay? If we talk first about the capsule, it's got a capsule surrounding it. Now this is different than what you talked about in terms of the kidney sitting in an adipose capsule. Retroperitoneal, there's a lot of adipose surrounding the kidneys and the adrenal glands. Rather than calling that an adipose capsule, if I could name it, I'd call it the adipose pillow. Okay? Basically wrapped in a pillow to protect the fine, delicate structures by cushioning for the fine, delicate vascular structures within the kidney. Adipose capsule is not a true capsule. That's why I call it an adipose pillow, an adipose cushion, something like that. But here's the actual capsule. You can see it staining very vividly here with the hemotoxin. And it's not a real tough, leathery type capsule like we see in other places. It does have slight trabeculae at a higher power magnification. You can see trabeculae starting to come in a little bit here, but there's no hint of any lobulation from it. Okay? No lobes or anything like that provided here by trabeculae coming off of this capsule. Now, when we look at it here, that capsule I point out as well, there's also reticular fibers here, reticular meshwork to help suspend these cells, particularly within the adrenal cortex. All right, the adrenal cortex, once again, cortex and medulla. Middle part's the medulla. We're going to see that's nervous tissue. The outer part here is the cortex. All right, and I tell you what, rather than my rushing through this to get done in 10 minutes, let's go and take our 50-minute break. Our 10-minute break after 50.